Welcome to our exploration of the diversity of chordates. Today, we are diving into the world of Osteichthyes, commonly known as the bony fishes. Imagine trying to build a submarine out of soft rubber. It simply would not withstand the crushing pressures of the deep ocean. Nature solved this engineering problem by developing a rigid, calcified skeleton. This evolutionary leap not only allowed fishes to master the aquatic realm, but also laid the structural foundation for all terrestrial life, including us. Let us begin our journey into their incredible biology.
To truly appreciate these creatures, we must first understand what sets them apart structurally. As we just mentioned with our submarine analogy, the defining characteristic of Osteichthyes is their skeleton. Unlike sharks and rays, which rely entirely on flexible cartilage, bony fishes possess an endoskeleton made of hard, calcified bone. If you have ever eaten a salmon and had to pick out the sharp bones, you have experienced this firsthand. This rigid framework is crucial because it provides superior structural support for powerful muscles, allowing these fishes to achieve remarkable feats of speed and agility in the water. Now that we know they have bony skeletons, let us look at their formal classification. The word Osteichthyes literally translates from Greek as bone fish, but their skeleton is just the beginning. One of their most vital anatomical innovations is the operculum. This is a hard, bony flap that covers and protects their delicate gills. More importantly, the operculum acts as a pump. By opening and closing it, bony fishes can actively draw water over their gills to extract oxygen. This means they can hover in place and still breathe, unlike many sharks that must constantly swim to avoid suffocating.
Beyond their internal skeleton and gills, their exterior is equally specialized. Their bodies are typically covered in overlapping, flexible scales, known as cycloid or ctenoid scales. These act like a suit of armor that protects them from predators while remaining flexible enough for swift swimming. This combination of traits has made them incredibly successful. In fact, they are the largest and most diverse class of vertebrates on the planet, with over 28,000 living species. Even more astonishing, a specific group called the lobe-finned fishes eventually developed limb-like fins, crawled onto land, and gave rise to all amphibians, reptiles, birds, and humans. Let us visualize these incredible adaptations we have been discussing. On your screen is a cross-sectional diagram of a typical bony fish. Notice the water entry at the mouth and the exit at the operculum, illustrating the active pumping mechanism for respiration. Along the exterior, you can see the overlapping scales providing flexible armor. Inside the cutaway, we see the rigid bony skeleton extending through the tail, providing that crucial structural support. You will also notice a large empty cavity labeled the swim bladder. This specific organ is an absolute marvel of biological engineering, and it is what we will explore next. Seeing the swim bladder in our diagram brings us to one of the most fascinating aspects of bony fish biology. Imagine having a built-in hot air balloon inside your body. That is exactly what the swim bladder is. It is a specialized gas-filled organ that allows the fish to achieve neutral buoyancy. This means the fish neither sinks to the bottom nor floats to the surface. It simply hovers effortlessly in the water column, saving massive amounts of energy. This is a huge advantage over sharks, which must constantly swim to generate lift. This hovering ability is a perfect biological application of Archimedes' principle, which deals with buoyant forces. To understand how the fish controls its depth,
we must look at the physics behind Archimedes' principle. The formula on screen shows that the upward buoyant force, FB, is equal to the density of the water, rho W, multiplied by the volume of the fish, V, and the acceleration due to gravity, G. The buoyant force is measured in newtons. The water density is in kilograms per cubic meter, and the volume is in cubic meters. When a fish wants to rise, it secretes gas into its swim bladder. This increases its total volume, V. Because the volume increases while the mass stays the same, the upward buoyant force becomes stronger, lifting the fish toward the surface.
Now that we have our formula, let us see how a real fish uses it. Suppose we have a freshwater bass with a mass of 2.0 kilograms, swimming in water with a density of 1,000 kilograms per cubic meter. We want to find the exact volume the fish must maintain to hover perfectly still. To achieve this neutral buoyancy, the upward buoyant force must perfectly balance the downward pull of gravity. We set the two forces equal to each other and plug in our numbers. Multiplying the density, volume, and gravity on the left and mass and gravity on the right, we solve for V. The result is 0.002 cubic meters, which is exactly 2 liters. The fish must adjust its swim bladder to maintain this exact total body volume. Having solved the problem with specific numbers, let us look deeper at the algebra to uncover a fascinating biological truth. We are going to derive the general condition for neutral buoyancy. We start exactly as we did in the example, by stating that the upward buoyant force must equal the downward gravitational force. We then substitute our formula for buoyant force on the left side and the standard formula for weight, mass times gravity, on the right side. This gives us the combined equation, where density times volume times gravity equals mass times gravity. This setup reveals something very interesting about the role of gravity, which we will resolve in the next step. Continuing from our setup, look closely at the equation. The acceleration due to gravity, g, is present on both sides. Mathematically, we can divide both sides by g to cancel it out completely. This leaves us with a beautifully simple relationship. The density of water times the volume of the fish equals the mass of the fish. Rearranging to solve for volume, we get volume equals mass divided by water density. This mathematical proof tells us something incredible. Because gravity cancels out, a bony fish's ability to achieve neutral buoyancy is completely independent of the planet's gravity. It depends entirely on the relationship between its mass, its volume, and the density of the fluid it swims in. We have seen how bony fishes master gravity, but that brings us to another massive physical challenge they face: the chemistry of their environment. This is the process of osmoregulation, which is how they manage the salt and water balance in their bodies. For marine bony fishes, the ocean they swim in is significantly saltier than their own internal blood and tissues. Because nature seeks balance, this steep concentration gradient means that fresh water constantly wants to escape the fish's body through its gills and into the salty ocean via osmosis. Essentially, a fish in the ocean is in constant danger of dehydrating. To understand the sheer magnitude of this threat, we need to calculate the osmotic pressure. To quantify this dehydrating force, we use a principle from chemistry called the Van't Hoff equation. The formula is pi equals I times m times r times t. Here, the Greek letter pi represents the osmotic pressure in atmospheres. The variable I is the Van't Hoff factor,
which accounts for how salt breaks apart into two ions in water, so we use the number 2. M is the molarity, or salt concentration, of the fluid. R is the ideal gas constant, and T is the absolute temperature in Kelvin. If the molarity, M, increases, the osmotic pressure increases, meaning a stronger pull on the water. Let us use this to calculate exactly what a marine fish experiences. Let us calculate the pulling force exerted by the ocean. We're given that the ocean has a molarity of roughly 1.0 moles per liter, and we will use a standard temperature of 298 Kelvin. We want to find the external osmotic pressure. We take our Van 't Hoff equation and plug in our values. 2 for the ion factor, 1.0 for the ocean's molarity, 0.0821 for the gas constant, and 298 for the temperature. Multiplying these together gives us a massive 48.93 atmospheres of pressure. This is the external force trying to suck water out of the fish. Now we must find out how well the fish can hold on to its own water. The fish's internal blood has a much lower molarity of just 0.3 moles per liter. Using the exact same formula, we substitute 0.3 for the molarity. This gives an internal osmotic pressure of only 14.68 atmospheres. To find the net force acting on the fish, we subtract the internal pressure from the massive external ocean pressure. The result is a net difference of 34.25 atmospheres. This invisible, crushing force explains why marine bony fishes must constantly drink seawater and actively pump out excess salt through their gills just to survive. We have covered a tremendous amount of ground today. To summarize, the class Osteichthyes represents a monumental leap in vertebrate evolution, primarily due to their rigid, ossified skeletons that provide unmatched structural support. We explored how anatomical marvels like the operculum allow for efficient breathing, and how the swim bladder manipulates the physics of buoyancy to save energy. We also calculated the intense chemical challenges of osmoregulation that marine species battle every single day. By understanding these physical and chemical mechanics, we can truly appreciate why bony fishes are the undisputed masters of the water, and a testament to the power of evolutionary engineering. Great job following along with the math and concepts today.